Lovely. So there's two of us, and we haven't rehearsed it, so let's see how we get on, and we'll talk over each other a bit, I would imagine. So I'm Dr. Lydia Green. I'm a paediatric neurologist working in Leeds. I'm working as part of the North IWMD hub. As part of my clinical job and 3 hours, which are of magnitude, you'll imagine, I'm also doing a part-time PhD supervised by James. It's very part-time in definition. And I'm looking at the neurogenetics of leukodystrophies, and then how we can, from a genetics and research perspective, bring that into clinical practice. And for me, this is all about linking clinical experience with research genetics and putting those things together, which I think is sometimes tricky in the UK from a medical perspective. And hopefully we're going to talk about how we're starting that process today.
So I'm James Poulter. I'm a geneticist at the University of Leeds. So I'm purely research-based. I don't work for the NHS. I work for a university, so I'm an academic. And I've spent the last, actually I don't want to count how many years it's been, but working in the field of genetics and trying to link how variants in people's DNA kind of contribute to inherited disorders. So as Lydia said, we're working together now to try and link up the leukodystrophy and the National Service with the research and trying to kind of improve outcomes for patients by kind of working together.
And then what we're going to do today, hopefully, is just provide a quick crash course in genetics. And I know James has done a little bit of that this morning, but we'll talk a little bit about that. We're going to talk a bit about what the NHS whole genome sequencing service is now. So what can you get on the NHS? And then really the challenge for patients with rare disease. So things are better in whole genome for some groups of patients. Some things are more challenging, and we haven't really bottomed those out yet. So we've started a new service without having all those pathways in place. And so we're clawing back a little bit on that side. And then what we're going to do about it. So how do we actually put that into action and not just words? So first of all, what are we? So as humans, we are built of our DNA. We have building blocks, and that makes about 20,000 genes within all of us. All of us have spelling mistakes in our DNA. Most of them aren't in important things. They're a spelling mistake that changes a bit of your protein, but it doesn't change what your protein does. It doesn't change how your enzyme works, so it doesn't matter. And as families, we have family spelling mistakes that just handed down with us and don't cause any trouble. And we have de novo or new spelling mistakes that when the egg and the sperm come together in the womb, we make them as we are built. But when do those spelling mistakes matter? When they happen in an important gene, an important protein, an important enzyme that has an important job in the body. And it changes how that protein is built or how that enzyme works. So part of your body's functioning then can't do the job it needs. Now, those spelling mistakes are called variants in genes, they're called mutations in genes, and different people will call them different things, but essentially it means something is built the wrong way, and that affects how it works later down the line.
So, yeah, I mean, this is just a kind of, obviously, we have, I think I said this morning, about 10 trillion cells in our body. Each of them, in theory, has the same starting DNA in the nucleus. But as time goes by, as Lydia said, certain cells in the body use the genes differently, and different variants have different effects depending whereabouts in the genome they are, if they're in an important region or not in an important region. There's also lots of different types of genetic variants, and as we'll come on to in a minute, it's very difficult to look at some types of variants, particularly in a national capacity, so the whole genome sequencing that's done on the NHS kind of only looks at a subset of variants that it's able to do in its capacity. So those are kind of the kind of single nucleotide variants, those kind of spelling mistakes that we see. But there are also much bigger variants that we see that are just much more challenging to detect, and that's where a lot of the challenges are nowadays.
So I spend a lot of my time trying not to be grumpy about genetics at work, which is a really disillusioned place to be sometimes. So most testing now on the NHS is through whole genome sequencing. We can still do some single gene tests, so there are some conditions where I think this is what you have, and I'm going to test for that. Or I'm going to test for a couple of genes that cause something similar to that. But that's not for most of what we do. So most of what we do is whole genome sequencing. So we're going to get all the information from all of your genetics, and if you're a child, hopefully from your parents as well, and we're going to do a trio and look at all of you. But the misnomer is we're not going to look at all of that information. We're then going to look at panels of genes within that. So although we've got all of your genetic data, we're going to look at panels within that. So for our patients, we're going to look at your white matter panel. Now, the white matter panel has over 1,000 different genes on it. There are not over 1,000 different white matter disorders, But there are now lots of genes that cause white matter disorders, and loads are leukodystrophy associated, or you've got a couple of fluffy white matter bits, but it's not a primary white matter disorder. But you can imagine we're now going for over 1,000 genes just in white matter. Sometimes that can be tricky to understand. And hopefully we're going to look at not just your white matter genes, because that's a really small place of your DNA to look at. So if you've got a white matter disorder, you've probably also got some learning. You might have some epilepsy. And we should be making sure that when we're starting to request these tests, we're requesting to look at all the relevant genes, because if we're going to spend 2 years getting a genetic result back, we should be looking properly. Now, what we don't do, and I think even your doctors won't always be aware of, is we're not looking at everything. So if I ask a question and say, please look at my white matter genes, but your answer doesn't lie in a white matter gene, I won't see it. So we're looking at a very specific place of your genome for an answer. Now, if your answer doesn't live within that gene panel, we will not find it. Despite having all the genetic information for you and your family, we're not going to pick that up. And we're only going to find, as James said, certain types of spelling mistakes. The whole genome is excellent. It improves the quality of the data that we get back, but it only finds certain types of mutations or variants. Now, there are techniques that can be used, but they're not used as standards to try and get these other bits. So unless we've got back and forth with our lab saying, this is what we need to look at, I need you to look at it in a special way to try and, that's not the standard way of looking, but if you could look in a different way for me, then we're not going to get all of the answers that we need from that test. So there are benefits to whole genome. The coverage of the information we get back is better than we did before. We get better quality, bigger volumes of information. There are benefits to panel testing because we only then have to look at a little bit of it. The NHS is stretched. If we said to every clinical scientist, look at all of this information, our turnaround times of 2 years would be 5 years and nobody would ever get an answer in enough time. So there are some benefits to doing it this way, but we have to understand the challenges about asking the right question, else it isn't a service that's going to work for our rare disease patients.
And the other thing that comes to mind is mitochondrial variants as well. I don't think the whole genome picks up the mitochondrial variants that well, I don't think. So obviously there's a lot of leukodystrophies that are associated with mitochondrial DNA mutations, and there's a separate test that's required to screen all of them. So it'll pick up all of the DNA that you find in the nucleus, but actually the DNA that we find in the mitochondria, which also contributes to disease, isn't actually detected by this test. And so you could spend a long time looking for things that are not even there in the first place.
If you're not looking in the right place, you're not going to find your right answer, and that's about who's asking for the test and asking you to do the right thing.
Go next.
Yeah.
So, this is a slide that I did just to kind of highlight. This is what the NHS pathway is for looking at variants. So they will take all of the variants that come back from the actual sequencing, and then they run it through a series of questions. And obviously the ideal is that you end up at the end with the one variant that we think is causing the disease. But that's not always the case. And if it doesn't, you might have a variant that's actually causing disease, but if it doesn't quite meet some criteria, and one of them is, is it in a gene that we're supposed to be looking in the first place, then it actually gets filtered out and puts into a different box that isn't then kind of passed forward and looked at. So from an NHS and from a diagnostic perspective, they have very high stringency of what actually gets reported back to Lydia and the other consultants. And so, we know that there are variants in those genomes that are not being picked up from an NHS perspective, just because of that. And there are good reasons for having those stringent criteria, but there's also, as Lydia said, needs to be a mechanism where we can kind of go back and interact with it.
So at the moment, if you look at James's flowchart, we will only be told about the tier 1 and the tier 2 variants. So we're not even told about the tier 3, never mind the other things that don't even get a tier. And that means we can't assess the relevance of those. So in the olden days, we used to get a report and sometimes it had 15 things on we don't understand. So they've taken the things we don't understand off. But in rare disease, what if they were important? If you don't tell me about them, I can't understand what they mean for you and your family, and that's really important and brings us really nicely onto the next bit about rare disease. So, rare diseases by definition affect 10 in 10,000 patients. Leukodystrophies are ultra-rare diseases. They affect less than 1 in every 10,000 patients, so that's less than 0.01% of you in the population. So that just means we know less about these diseases, we know less about the genes, we have less evidence from the current spelling mistakes we know. So genetic testing works on what we already know. So if we already know about that gene, we can look at it. Within that gene, we're looking for evidence that those same spelling mistakes, those same problems, have already been found in other families. But what if you've got a new spelling mistake in a gene we know about? There's no evidence for that. So they're not going to tell us about it. What if you've got a new gene being involved in a rare disease we've never seen before? That's not going to find it on our testing at the moment. And if you don't tell us about those things, we can't look at them. You will have spelling mistakes that are completely irrelevant to your clinical condition, but there may be one in there that's making the problem, but unless we know about them all, we can't try and investigate them further. So this is about doing further tests, not just saying, yep, there's a variant we don't understand, let's label that. It's saying, tell me about what we don't understand so I can look at it further, so we can work out if it's relevant. It might not be the answer, but unless I look at it, I can't tell you if it's the answer, and you continue on your diagnostic odyssey for years to come without having an answer. So I need to know that information at the start so that we can investigate it to see if it's relevant for you. Now, before whole genome sequencing, so at about 2017, we did exome sequencing in Leeds where we looked at our patients with rare diseases and leukodystrophies. We had our own gene panel. We grew that over time. Over 5 years, 20% of our diagnoses were in new disease genes, novel variants in new disease genes. So 20% of our cohort in that 5-year period will not be found by current NHS testing. And the downside is we don't currently have a pathway. If you are negative on NHS testing, there is nothing else I can do. There is no other NHS pathway to finding the diagnosis. You are stopped and you are stuck. So we need as a service to move that forwards, because as much as I want to be able to identify our more common conditions, move you through to trials, move you through to therapies, I actually really want to, alongside that, look at our 20, 30, 40% in some types of leukodystrophy, undiagnosed patients, because a diagnosis is important. Without a diagnosis, even if I can't cure you, even if I can't give you a treatment, without a diagnosis, you don't have closure. You don't have prognosis. You don't have someone sitting honestly and telling you, yeah, it might be the worst thing that you could be told, but it's important to be told that, because if you could never get to that point, how can you live your life and deal with the rest of it? You need that first step to be able to absorb that and then go, okay, what's life living with this? We can do this. We're a family, we've been doing it for years, but we can do this and we can focus on quality. But we need to give you that first step and confidence of diagnosis. So we need to now move on from NHS into the research spectrum to be able to provide a pathway for you.
So in the research setting, we have a little bit more flexibility on what we can do. Obviously, the NHS is stretched and they have very strict criteria. Now, what's a really good thing about the NHS genetic testing is that once they've finished with that genomic data, it's put into a secure research environment, a digital environment, where researchers who have applied and got permission can go in and actually look at that data again. So what we can do is actually take that raw data that's already been through the NHS process, and we can look at it in all of those different ways and start digging and digging around and pulling apart and looking at the different types of variants to see if actually we can spot the variant using our criteria and using our pipelines. Now, again, there's a whole kind of level of kind of things we need to meet. So again, in the first instance, we need to get access to that data. And that's one of the biggest challenges at the moment, is kind of waiting for that data to be put into the research environment. And then we do just a simple, has anything obvious been missed? And sometimes it is something really obvious that's been missed, and these things happen because they're screening and testing so many patients at the same time. So we can go in and see if there's anything obvious that we can pick out first. And then, as I say, we can use all these extra tools. And then what's really key is actually a back and forth conversation with, between the clinical teams, between the research teams, and having these multidisciplinary team meetings where, we update information. And a lot of the problems we're finding with the genetic testing is that the information, the phenotypes, the symptoms are put in incorrectly or into the system in the first place, so that the analysis that's done by the NHS is based on incorrect information. So that's why a lot of this is being missed. If we can go back in and update that information so that it's correct, actually the variant might be there all along, and it might kind of drop out and be obvious once you kind of updated that information. There's obviously kind of situations where there are new things and there's things we don't understand, and again, the NHS doesn't really have the capacity to look at them, but we in the research labs do. So we can take variants that we don't understand and try and test them, try and do something in a lab that says, if we introduce this variant into this gene, does it stop it working? Does it do something that makes it not do what it's supposed to do. And as Lydia said, these are the crucial genes that we need them to function in our bodies. And so if we can show when we have this variant in a gene, it doesn't work anymore, then that's a lot of evidence that says, actually, if this was in a human, then this protein, this gene wouldn't work anymore. And that's how we can try and link it in with these phenotypes. And we can also look in fish models. We can see what happens to the myelin in a zebrafish when we introduce this variant. So there's lots of things and evidence that we can build in a research lab that can contribute and kind of feed into this pipeline. And then hopefully we can get a diagnosis at that point. And I guess that's really where the clinical team comes in, and that communication is really helpful.
And I think it's only this combination or collaboration between the clinical team, so we're getting the right phenotype at the start. So what does this patient actually look like? What does their scan mean? And where should we look? The lab experts looking at the science, and then the research network of fish, flies, and all of those other things proving. Because we still need to prove what we're finding is causing that patient's problem. Because if we're labeling the wrong thing, misdiagnosis does not help anyone. So we still need to have a high level of accountability in what we do. But we've got families recently, we had a gene in 2016 we've had for ages in our service. We've now just about to publish a series of 19 patients in 13 families from all around the world, all of whom have had this spelling mistake identified over the last 10 years, but haven't yet been able to have a label. But only by bringing that patient cohort together can we actually say, this gene is causing this problem, this is what these patients look like, this is what these scan looks like. And by publishing that, we then open that up to other people who then can make that diagnosis going forwards. But in rare diseases, that's how it has to work. We have to have the collaboration between the clinician, the lab scientist, and the research network to prove it, and then we can bring that back to the NHS and say, okay, we've got something for you now, add that to your system. And then for those patient groups, once we know about it, we can start looking at it from a treatment pathway. That's a bit down the line, but until we know, we can't even move on to that bit.
So, yeah, I mean, I think what's the key thing here is, like Lydia said, is this collaboration. It's kind of going this back and forth and making sure that the information in the first place is correct, because, a lot of genetics, to find that one variant, that needle in a haystack, we need to be looking in the right place in the first place, and we need to have the right information to find there. It's like a treasure map. If that X is in the wrong place on the map, you're never going to find the treasure. And it's exactly the same in genetics. We need to have the right information and the right clinical information to really kind of pinpoint in on what we're trying to find. And it's not going to solve everyone. We're still not going to pick up everything. And there are new technologies coming along that are helping us to find things that we've never been able to see before. But again, it's one of those situations where being able to find the ones that we can find, and then the ones that remain unsolved, we can then prioritize them for the new technologies that are coming along and make sure that they're getting the best techniques used on their DNA that we can.
And I think the final summary is, so we've talked a lot of words, but when can we actually put this into action? So from the start of next year, we're going to be piloting this research pathway for all of our genome negative patients in the north of the IWMD network. The reason we're starting there is because they're patients that have been through our service that we know and we've seen. So I will have seen every single one of those patients before we put them through the pathway, and that helps us be able to interpret that genetic data better, because we need to evaluate, can we do this successfully with the group we know, so that we can then roll that out for the wider network. So my aim is that nationally, if you've had a white matter service contact and you're undiagnosed, you can then come automatically down our pathway to go from an undiagnosed NHS test to a research pathway that's set up and ready to take you and move forwards in a sensible way. So what we can do now is look at our local network. We can do that with the money we've got. What we're going to need to do, if we can prove that works, is then look at funding to take that for the wider national network going forwards. And I'm hopeful that in the next 12 to 18 months, that's where we are, and that next time, or maybe in 2 years at this meeting, we're feeding back the findings of that national network of research and gene discovery that's making a difference for our patients.
Thank you.
So whole exome and whole, so an exome is the protein coding bits of your DNA. So when we do a whole exome, we get the information from all of the protein coding bits and a little bit outside of that. When we do whole genome, we get everything. So there's loads of stuff in the middle that we don't really know what that does. Now we're still in whole genome. We're not looking at that stuff in the middle because we don't understand it all. We're getting a little bit better, but there's still bits we don't even look at. So we get it all, but we don't look at it. But the quality of the data that we get when we do whole genome, even if we just look an exome from a genome, the quality of that exome data is better. So it just gives us better data to work with generally, even if we're not looking at much different stuff. The amount of data is vastly, yeah.
It does take longer, but actually, like Lydia says, actually, if we in terms of the processing of the data takes longer, but actually the looking at the data is not that much different.
A lot of it's automated, so there's a lot of pipelines you build up. So you stick the stuff into the computer at the start, and that's your genetic information, then your clinical information, and it then automates it, how it goes through, and then we need to pick it up and